Hello everyone, welcome back to the channel. In today's lecture, we will be learning about sensation of smell, covering the structure and function of olfactory receptors, the olfactory pathway, and the role of vomeronasal organ, olfactory transduction, also the types of odors, adaptation, and common clinical abnormalities. So let's get started. The sensation of smell, also known as olfaction, is the process of detecting volatile chemical substances that are suspended in the air. It is categorized as one of the two chemical senses, the other being taste. Evolutionarily, olfaction is one of the most ancient senses, present even in the simplest of organisms. For a chemical to stimulate olfactory receptors, it must be volatile so it can reach the nasal cavity and usually both lipid soluble and water soluble to penetrate the mucus and bind the receptors. The sense of smell plays several vital roles. It enhances the perception of taste. In fact, what we call flavor is actually a combination of taste and smell. It helps us detect dangers such as smoke from a fire, leaking gas, or spoiled food. It has strong connections to the limbic system in the brain, linking smell directly to emotions and memory. That's why certain smells instantly bring back powerful memories. It also plays a subtle but important role in social interactions, possibly detecting pheromonal signals subconsciously. Even though human olfaction is not as developed as in some animals like dogs, it remains highly sensitive, capable of detecting thousands of different odorants. The olfactory receptors are highly specialized structures found in the olfactory mucosa, also known as olfactory epithelium, located at the roof of the nasal cavity. In humans, this sensory area covers about 5 square centimeters in each nostril. The receptors themselves are bipolar sensory neurons. At the apical end, they have long, non myelinated cilia that extend into a mucus layer. These cilia are critical because they contain the odorant receptor proteins that detect smell molecules. At the basal end, these neurons form unmyelinated axons. These axons bundle together to pass through the cibriform plate and form the olfactory nerve fibers known as cranial nerve 1. Supporting these neurons are two important cell types: supporting cells, which provide physical support and detoxify harmful substances, and basal cells, which are stem cells capable of regenerating new olfactory neurons. This regeneration is a unique feature as most neurons in the body do not regenerate. Bowman's gland found beneath the olfactory epithelium secrete mucus that plays several roles. It keeps the olfactory surface moist, it traps and dissolves odorant molecules, and it contains odorant binding proteins that enhance the capture of smell. An important point to remember: the olfactory receptors are directly exposed to the external environment, making them vulnerable to damage from infections, chemicals, physical trauma. So, fortunately, they have a rapid turnover, regenerating every 30 to 60 days, maintaining our sense of smell throughout the life. Now, let's focus on the vomeronasal organ, also known as Jacobson's organ, a fascinating but often overlooked part of the olfactory system. The vomeronasal organ, or VNO, is an accessory olfactory structure that specializes in detecting pheromones, chemical messengers involved in communication between individuals of the same species. In most animals, it is located bilaterally within the nasal septum, enclosed by cartilage. Structurally, the VNO is a small, fluid-filled tubular organ lined with specialized receptor cells connected to vomeronasal nerves. The VNO plays a critical role in mediating instinctive behaviors such as mating and sexual attraction, maternal recognition, offspring bonding, territorial behavior, and aggression. It detects non volatile chemical signals that do not travel freely through the air like normal smells. The signals picked up by the VNO are sent to the accessory olfactory bulb, which then projects directly to the amygdala and hypothalamus, regions controlling the emotion and hormonal responses. In many lower mammals like rodents and reptiles, the vomeronasal system is fully functional and essential for survival and reproduction. But in humans, the existence of vomeronasal pits is well established. But whether the human VNO is fully functional remains a topic of scientific debate. It may still play a subtle subconscious role in human chemical communication, but clear evidence is still being explored. Now let's carefully trace the olfactory pathway, the route through which smell information travels from the environment to the brain. Step 1 is detection. It all begins at the olfactory receptor, specialized bipolar neurons located in the olfactory mucosa. These neurons detect odorant molecules that dissolve in the nasal mucosa. Now step 2, formation of the olfactory nerve. The axons of the olfactory receptor cells bundle together to form the olfactory nerve, cranial nerve 1. These bundles pass through the tiny holes of the cribriform plate of the ethmoid bone, entering the nasal cavity. Understand? Step 3, synapse in the olfactory bulb. Upon reaching the olfactory bulb, these axons synapse with specialized structure called olfactory glomeruli. Here they interact with second order neurons, mainly the mitral cells and tuft cells. Step 4, formation of the olfactory tract. The axons of mitral and tuft cells converge to form the olfactory tract, a band of nerve fibers carrying processed olfactory signals. Step 5, division into olfactory striae. The olfactory tract splits into three striae. Lateral olfactory stria. This is major pathway sending signals to the primary olfactory cortex. Intermediate olfactory stria involved in feedback mechanisms and modulation of smell. Medial olfactory stria, some fibers cross over to the opposite olfactory bulb for bilateral integration. Now step 6, termination in primary olfactory cortex, the final destination, which includes the prepiform cortex, important for basic odor perception. The anterior olfactory nucleus modulates olfactory signals. The entorhinal cortex, which links the smell to the memory. The amygdala, which associates smells with emotion. And the periamygdaloid complex. Unlike other senses like vision or touch, the olfactory signals bypass the thalamus initially. They directly reach the olfactory cortex first, highlighting the primitive and urgent nature of the sense of smell. Now let's explore the fascinating process of olfactory transduction. That is how the chemical smell stimulus gets converted into an electrical nerve signals that the brain can understand. So here is the step-by-step mechanism. Step 1 is the odorant molecule must first dissolve in the mucus that lines the nasal cavity, which is secreted by the Bowman's gland. Step 2, once dissolved, the odorant binds to the specific receptor protein located on the cilia of the olfactory receptor cells. Each receptor is specific to a range of related odorant molecules. Step 3, binding of the odorant activates a G protein, specifically called the gall protein in the olfactory cells. Step 4, the activated gall protein stimulates an enzyme called adenylyl cyclase located inside the cell membrane. Step 5, adenylyl cyclase then increases the level of cyclic AMP inside the receptor cell. Now step 6, the newly formed cyclic AMP molecules open cyclic nucleotide gated or CNG ion channels. Step 7, this allows influx of sodium and calcium ions into the cell, leading to depolarization of the receptor membrane. Step 8, if this depolarization reaches the threshold level, a receptor potential is generated. This leads to initiation of an action potential which travels along the olfactory nerve fibers to the olfactory bulb. Now, amplification mechanism. An interesting point is that a single odorant molecule can activate multiple G proteins, thereby amplifying the signal significantly. This explains why the olfactory system is so sensitive, able to detect even minute concentrations of odors. Role of calcium. Calcium influx not only helps in depolarization but also plays a role in adaptation, meaning the receptor becomes less responsive to continuous stimulation, helping prevent sensory overload. Olfactory transduction is characterized by high sensitivity and rapid signal processing, allowing us to respond quickly to important smells in our environment. So, the sense of smell is a remarkable example of how chemical information is transformed into electrical language the brain can understand in a fraction of a second. Now, let's talk about the classification of odors. So, odor perception is based on the interaction between odorant molecules and the different combinations of olfactory receptors. Interestingly, there is no single universal classification system, but many practical groupings have been proposed based on common sensory characteristics. Here are some commonly recognized categories. Floral odors. These are sweet and pleasant smells typically associated with flowers. Examples include rose, jasmine, and lavender. Fruity odors. These smells are fresh, sweet, and sometimes slightly acidic. Examples include apple, banana, strawberry. Spicy odors. These have a sharp, warm, and aromatic profile. Common examples are clove, cinnamon, and pepper. Resinous odors, heavy sticky scents that resemble plant resins or sticky trees. Examples include pine, balsam, and tar. Burnt odors, smoky or charred smells. Examples include roasted coffee, roasted meat, and burnt wood. Garlic like sulfury odors. These are strong pungent smells, mainly from sulfur containing compounds. Examples are garlic, onion, and rotten eggs. Additionally, some other categories have been proposed. Putrid odors associated with decay and decomposition, like rotten meat. Camphoraceous odor, sharp, cooling, and medicinal, like camphor or menthol. A few important points to remember. Most natural smells are not pure. They are mixture of multiple odor types. Our olfactory system performs complex coding, detecting combinations of receptors activated by a single smell, creating unique perception profiles. Individual genetic variation also influences how people perceive and respond to different odors. So, while these categories help describe smells, the reality is so far more complex and dynamic than simple labels. Now let's discuss an important aspect of olfaction, the threshold for olfactory sensation. The threshold refers to minimum concentration of an odorant needed to trigger a detectable smell sensation. A remarkable feature of the olfactory system is its extremely high sensitivity. Humans are capable of detecting some chemicals at incredibly low concentration. For example, methyl mercaptan, a chemical often added to natural gas for leak detection, can be sensed by the human nose at concentrations as low as 0.002 parts per million or ppm. That's like detecting a few molecules of a substance in a full room of air. Truly amazing. However, it's important to note that different odorants have different threshold. Some smells, like floral fragrance, are detected by very low concentration, while others, like some chemicals vapors, require higher amounts. Several factors influence an individual's odor detection threshold. Genetic differences. Some individuals are naturally less sensitive or even completely insensitive, means anosmic, to specific smells. Physiological states, conditions like pregnancy can make women hypersensitive to certain odors. Age, olfactory sensitivity tends to decrease with age, explaining why older adults may notice smells less accurately. Health conditions, infections, allergies, smoking, trauma can blunt the sense of smell. From a practical perspective, this high sensitivity is essential for safety. Detecting gas leaks, smoke, or spoiled food early can be life-saving. It also enhances the enjoyment of flavors, as smell is a major component of how we perceive the taste of food. Now, let's explore the important concept adaptation in smell, also known as olfactory adaptation. So, adaptation simply means that after continuous exposure to a particular odor, our sensitivity to that smell decreases over time. One unique feature of our olfactory system is that it is phasic, which means it responds most strongly to new or changing smells but becomes less responsive to smells that are constant. In fact, research shows that within just 1 second of continuous exposure, there is already about 50% decrease in the sensory response. And with prolonged exposure, we can even reach complete adaptation, where we no longer notice the odor at all. How does this happen? Adaptation involves changes at two main levels. First one, receptor level adaptation. At the receptor level, continuous stimulation leads to a buildup of calcium ions inside the olfactory receptor cells. This triggers a feedback mechanism that inhibits ion channels, making the receptor less sensitive to the odorant. Second, central adaptation. At higher brain centers like the olfactory bulb and olfactory cortex, inhibitory neurons such as granule cells actively suppress the repetitive signals so that the brain does not keep paying attention to an odor that no longer relevant. We experience olfactory adaptation every day. For example, after applying perfume, you notice it strongly at first, but after few minutes, you stop noticing it. When you enter a room, you may detect its scent immediately, but after staying for a while, the smell seems to disappear. Why is olfactory adaptation important? It prevents sensory overload from constant background odors, and it allows our nervous system to stay alert to new smells like smoke, gas leaks, or fresh food, which might be critical for surviving. In short, olfactory adaptation is a brilliant way our body balances awareness and efficiency and helping us focus on what's new and important in our environment. Now let's look at some important clinical disorders related to our sense of smell. These are conditions where ability to detect, recognize, or interpret odors is impaired. First, anosmia. It refers to the complete loss of sense of smell. It can occur due to upper respiratory tract infections like common colds, sinusitis, nasal obstruction from polyps or tumors blocking the nasal passage. Head trauma, especially fractures of the cribriform plate, can damage olfactory nerve fibers. Neurodegenerative diseases like Parkinson's disease, Alzheimer's disease. More recently, viral infections like COVID-19 have become notable cause of temporary or persistent anosmia. Next, hyposmia. It is the partial loss or reduced sensitivity to the smell. It can result from aging, which naturally reduces the number of functionally active olfactory receptors, chronic rhinitis or sinus infections that cause prolonged mucosal inflammation, smoking, which damages the nasal mucosa and impairs receptor function over time. Then hyperosmia. Hyperosmia is when a person experiences an excessive or heightened sense of smell. This can be seen in hormonal changes such as during pregnancy or adrenal insufficiency, certain neurological conditions where sensory processing becomes hypersensitive. Two more rare but interesting abnormalities are parosmia, where the perception of smells is distorted. For example, something that usually smells pleasant may now smell foul or rotten. Phantosmia, where a person perceives smells that are not actually present. This condition can be associated with migraines, temporal lobe epilepsy, or psychiatric disorders. Clinical importance. Loss or distortion of smell is not just a sensory annoyance. It has a profound impact on quality of life. It can lead to nutritional issues because flavor perception is diminished, safety risk, inability to detect gas leaks or smoke, emotional distress, and depression due to the intimate link between smell, emotion, and memories. Thus, recognizing and evaluating olfactory abnormalities is crucial both in clinical diagnosis and patient care. All right, let's quickly wrap up what we learned today. The sensation of smell or olfaction is a powerful chemical sense that detects volatile substances in our environment. Specialized olfactory receptors transmit signals through the olfactory bulb and tract directly to the brain without first passing through the thalamus, a unique feature. Olfactory transduction transforms chemical signals into electrical impulses using G protein mechanisms and cyclic AMP. Our olfactory system is designed for rapid adaptation, helping us stay alert to new and important smells. Finally, abnormalities like anosmia, hyposmia, hyperosmia can seriously affect quality of life and sometimes indicate deeper health issues. If you found this lecture helpful, please support our channel. This is a free educational platform aimed at making complex concepts simple and accessible to everyone. Your likes, shares, and subscriptions are truly help us grow and continue creating quality educational content. So don't forget to subscribe and turn on the bell icon so you never miss a future lecture. Thank you so much for your support and see you in the next video.